please welcome to talk about whether microbes have immune systems, Professor Robin May.
Thank you so much, Chris, and thank you all for coming or for joining us online. It's great to see you here. It's great to be back for this, the third year, and the first lecture in the new series on immunology. And what I want to do in this series is roam very widely through immunology, through the kind of immunology that is hopefully familiar to you, from vaccinations, from infections, to things that maybe are much less familiar, immunology of plants, of microbes, and in the future of computers too. But today is all about the kind of immunology that you might not have thought about for quite a long time, which is the immunology happening in cells that we can't even see. So the question I want to ask is, do microbes have immune systems? And Chris and I were saying beforehand that generally as academics, we think that whenever anyone poses a question in the title, the answer is almost always no. Except on this occasion, it's yes, otherwise it'd be a really short lecture. So I want to start off by asking you to reflect on the fact that today, all of you in this room will have come close to death. And at the moment, actually, unfortunately, some of you are dangerously close to death as we speak. The good news, I hope, he's confidently going out here, is that you will still all be here at the end of this lecture. If not, that would be very, very bad for us, and certainly memorable in all the ways we don't want it. So hopefully you'll make it through to the end. But the dicing with death that has happened to you on multiple occasions today has not resulted in your death because of your immune system. And I want to start with an example of that. I am sure that many of you today will have given yourselves a tiny cut, perhaps even a cut you haven't noticed. You've scuffed your finger on a grain of sand or a paper cut or something like that. Unless that cut was done with a sterile instrument in a sterile environment, like you're an operating surgeon, for example, you will have introduced into that cut bacteria, perhaps fungi, parasites maybe, things that, if left unchallenged, would undoubtedly spread through your body and might even kill you. And yet you're not, hopefully, feeling any worse for wear because your immune system has protected you. And let me show you what I mean by that. So this is an example of exactly that kind of reaction. This happens to be in a zebrafish rather than a person, but the principle is exactly the same. So this fish has experienced a tiny cut to its tail, that's this bit down here, too small actually to be seen without a microscope, and probably too small for the fish to even notice. But even though the fish hasn't noticed, its immune system definitely has. And what you will see here is pouring out of this nearby blood vessel are individual white blood cells that are racing towards the site of that wound. And although you can't see it, what they're doing here is gobbling up all the bacteria that have come into that wound, destroying them, protecting the body from that infection, and actually also signaling to the underlying tissue that it's time to start repairing that wound and stop other things coming in. This process is happening in at least some of you, perhaps all of you, right at the moment. Okay? You will have cut yourself in a microscopic way on the way in here, perhaps holding the door handle. You might have breathed things into your lung, which are trying to burrow into your lung and cause an infection, and a similar process is happening there. You might even have eaten something slightly dubious on the tube on the way here, and in your intestine, right at the moment, a very similar process is protecting you. So you are kept alive because of your immune system. And later on in the series, we're going to explore exactly what that means and exactly how that process works. But today, I want you to think not about your own immune system, but the immune systems that we almost never think of, those of much smaller individuals, and in particular, those of single-celled organisms. So I want us to focus mostly on the immune system of bacteria. And here is a classical, typical bacteria. This is E. coli, bacteria you can find in your intestines. It's the kind of workhorse of laboratory experiments around the world. Like pretty much all bacteria, it is a single-celled organism, microscopic. It's about 1 micrometer long, so about 1,000th of a millimeter, and it is pretty simple on a biological level. And you might think that an organism this simple doesn't need any form of immunity, but you would be wrong. Because just like ourselves, bacteria have diseases and potentially fatal diseases. So if you're an E. coli, this is your equivalent of sort of some kind of poster from a horror movie. This is a bacteriophage. It is a virus that is specialized to infect and sometimes destroy bacteria. So if you're an E. coli, this is a pretty threatening thing to do. It looks, for all the world, like it's been designed or created. It looks very kind of machinery-like, but actually it's a natural biological entity. And so if this is your kind of horror movie picture as an E. coli, this is the horror movie itself. This is one of your fellow E. coli being kind of eaten alive by bacteriophages. So here is the E. coli, the big grey thing. And what you can see, these little bright white dots, are these bacteriophages, these viruses, latched onto the surface, injecting into the bacteria their own DNA in order to replicate the virus and ultimately destroy that bacterial host. So this is definitely, if you're an E. coli, this is the kind of thing that keeps you awake at night in horror. So let's think a bit about how that process works. So here is a sort of diagram. Here's my sketched E. coli. So what typically happens is that your bacteriophage will encounter that bacteria. They vary. Some bacteriophages are very indiscriminate. They'll attack lots of different bacteria. Some are incredibly specific, targeting only a single species, or sometimes even just a particular strain of a particular species. And what they do, though, when they recognize their host, they latch onto the surface, and then they inject, actually in a mechanism that is a bit like a kind of mechanical ratchet, they inject their own DNA into that bacterial cell. And then that DNA hijacks the bacteria's own machinery to create copies of itself. So instead of the bacterial cellular machinery using the bacteria's own genome, it starts reading the viral genome and producing viral proteins. And those proteins assemble new viruses. And typically for many of these viruses, the ultimate outcome is you get more and more viruses, and eventually they lyse the bacteria, so they're called bacteriolytic viruses, and they explode out and go off to spread new infections. So if you're a bacteria, this is really bad news. And for any of you who came to any of the lectures last year on evolution, you will know that anything which kills things at a high frequency is a very strong evolutionary pressure. So you might expect that over the billions of years we've had bacteria and bacteriophages, as far as we know, these probably evolved around the same time as these. You might expect that evolution has driven a mechanism to try and prevent this from happening, and you would be right. And in fact, there are multiple mechanisms of bacterial immunity. One of the earliest to be discovered is a process that is actually kind of quite amazing in its simplicity. And it is based on the fact that a bacteriophage, a bacterial virus, will inject DNA into bacteria. So you can't discriminate the DNA itself, because bacteria have DNA too. But what you can discriminate is the letters of that DNA. Because as you all know, hopefully, DNA is written in single letters, and essentially it's a recipe book for producing things like protein. So you might have yourself a piece of DNA that says blue eyes or brown eyes or whatever. And they're subtly different. So for example, here's a string of letters that might be on that DNA that's come in from a bacteriophage. And many of those letters will be the same, and as words, DNA words will be the same as the words in a bacteria. But if you can identify a word that is specific to the virus and not present in your own genome, then as a bacteria, you can recognize the fact that you're being attacked. So let's imagine there's a little sequence here that the bacteria doesn't have in its genome, but the virus does. That is your hallmark to do something about it. And what bacteria have done is evolved a system called restriction enzyme digestion, which relies on enzymes, proteins, bacterial proteins, that recognize sequences of DNA not present in their own bacteria. And when they recognize it, they rapidly bind it, and they chew up the DNA that's associated with that sequence. So this is a very straightforward system. Essentially what you do is you say, I'm going to write my own DNA code to create a bacteria, and I'm going to avoid using certain combinations of letters, certain words. And I'm going to then create enzymes that recognize that missing sequence and digest it. And so if anything comes into me with that missing sequence, I will be protected. This is a very effective method of fighting off viruses. It's also a method that has become unbelievably important to us as humans, because this is the basis of the very first steps towards genetic engineering. Because what people did in the 60s, 70s, and 80s when they discovered this process is they realized that this enzyme is protecting bacteria by recognizing highly specific sequences of DNA. So what you can do if you're a biotechnologist is you can create those sequences, and now what you have is a pair of molecular scissors, if you like. There are other enzymes that will also stick DNA together, so now you have the components that you need to do genetic engineering, because you can engineer bits of DNA that have a particular label that says, cut me here, and then you can stick different bits together. And this is precisely how people produce all those fantastic things you see in the literature about glow-in-the-dark fish or antibiotic-resistant things or pesticide-resistant plants or whatever it is, by doing this process, harnessing this process of bacterial immunity. So that is one really powerful immune system in bacteria, and actually for a long time we thought that was probably the main and the only one. But then, just about a decade or two ago, actually people discovered there is a second immune system present in many bacteria. And this is, again, one that has turned out to be pretty revolutionary in its science. And it works like this. So the problem is, if you're a bacterium and you have a restriction enzyme system, that is great at the moment you're attacked by a virus. You've chewed it all up, fine, protected yourself. But you have not gained anything. Tomorrow, when a different virus comes, you've got to do it all again. You've got no memory, no immune memory. So what some bacteria, many bacteria have evolved is a system that provides them with immune memory. And what they do in that system is they take a copy of the foreign viral DNA that has entered their cell and they store it away as a memory of the pathogen they've encountered. It's a little bit like when you have a vaccination. You've seen a foreign entity, a bacteria or a virus, and your immune system has remembered what it looks like, ready to reactivate next time. So bacteria do that too. And so this is how it works. These are fragments of viral DNA up here in the corner from a virus that's been fought off. The bacteria integrates those into its own genome at a particular site that is flanked by these orange sequences here. And that site essentially says, this is my memory bank. Everything in this bit of my genome is not mine, it's come from a virus. And so if I see this again, I know I'm in trouble. And this process is one that you might have heard of called CRISPR. And it's a terrible name in the sense that it's for clustered, regularly interspaced palindromic regions. It's a good one for Scrabble. And essentially, this bit is quite straightforward. They're a cluster because they're all in the same place as the genome. They're regular. Actually, they're very nice and sequential. They're interspaced, obviously. They're palindromic, which means that the DNA sequence reads the same one way as it does the other. And actually, that has a very practical advantage for us as scientists because it means that computer-based programs can identify these quite easily because you tell it to look through the millions and millions and millions of DNA sequences for something that is clustered, regularly interspaced, and palindromic, and pretty rapidly you come down to candidate areas that look like this. So CRISPR is a really remarkable way for bacteria to remember an infection and deal with it, because now you have, as a bacterium, a little kind of copy of the stuff you've seen. What you do with that copy is you permanently produce, at low level, a copy of that memory, if you like, in a molecule called mRNA. This is like a version, very closely chemically related to DNA, but slightly different, but is retaining the sequence specificity. So you're peeling that off, if you like, as a bit like a kind of carbon copy for those people who remember carbon copies in the old days that you could take, or a photocopy today. And then you use this green template to compare to every other piece of DNA you ever encounter in your cell. So now, if a bacteriophage tries to infect you, it will bind, it will inject its DNA, that's all hunky-dory. But now, you've got a system to compare immediately. Without needing to read the sequence or anything else, you can just check, does this bit of DNA that's come in match this thing I'm producing? And if it does, you know that that DNA is coming from a virus. And that engages a system called the Cas9 system, a cluster of enzymes which come in and then degrade the DNA that's coming in. So you are primed and ready to immediately destroy anything foreign that's come in. Very, very effective. Also has turned out to be an incredibly important machinery, if you like, for human ingenuity. Because as soon as we kind of discovered this, it had an immediate application in many of the places we use microbes. So, for example, if you think about yogurt production, right? If you're, I don't know, one of the big yogurt manufacturers, you have these massive vats bubbling away yogurt. They're full of good bacteria, the bacteria that make your yogurt. You really, really don't want those bacteria to get a viral infection because then you've wiped out thousands of liters of production. So you can make sure that your bacteria are immunized, if you like, by having the CRISPR sequence for the core viruses they might get. So you're vaccinating your yogurt stock before it goes into fermentation. It's also harnessed a lot of interest in how we might use this kind of bacteria-virus interplay for medical benefit. So, for example, could we produce mixtures of bacteria that are resistant to bacteriophages, good bacteria, that you could then use to help harness your microbiome? So could you kind of protect people's gut flora by having really resistant bacteria? Or conversely, could you inactivate this resistance system to make bacteria vulnerable to viruses and then kill bad bacteria using those viruses? So could we give people, instead of antibiotics, could you give them bacteriophages to wipe out whatever infection they have? But most fundamentally, in the last few years, we have started to use, in the same way that we use those restriction enzymes, we have started to use CRISPR for all sorts of interesting technological applications. And the big one, the big breakthrough, if you like, was the understanding that you can tweak this system slightly and start to do all sorts of interesting things. And the big tweak that people made was changing those enzymes that they normally use to break down DNA into ones that can slightly modify it instead. So what we can now do in labs is you can use this CRISPR system and you can hijack, if you like, this copy and put in a sequence you're interested in. So you say, I want you to match this particular gene. It will work just like it does in a bacteria. So if it recognizes a piece of DNA with that gene that you've programmed, if you like, it will bind to it and it will call in the same protein complex. But if you now tweak that protein slightly, so instead of chewing up your DNA, it just puts a tiny nick in it, a tiny single letter change, you can now use this machinery to selectively edit, to change the sequence of that DNA. And that is exactly the process that has been harnessed to enormous power in genetic technology. So we now use CRISPR in labs all around the world to do things like, for example, engineering particular model organisms, cells or mice or fish or whatever, to have human versions of a gene that we're interested in for a disease. We use it to create crops that might be resistant to herbicides or to particular pathogens by highly selectively editing individual genes. And this is the basis, for example, in the UK, of the Precision Breeding Act, which is a new change that will bring on board crops that are modified in this kind of way to have all sorts of traits in terms of beneficial nutrition or greater crop productivity. And most excitingly, for me at least, as someone who's come from this world, is the fact that it is now actually starting to impact directly on medicine. And you might be aware that last year, the UK became the first country in the world to authorize this as a medicine for people. And in this particular case, what we have licensed it for is for the treatment of 2 blood disorders, like beta thalassemia. So these are genetic disorders in which people have a mutation in genes that are critical for red blood cell function. And these people have a lifelong disability and illness and require regular treatment. It's a pretty straightforward mutation, relatively simple. So we know what you'd have to do to correct that mutation back, but we have never before had the tools to do it, but now we do. So now what people can, if you have this condition, what you can have is an injectable form of this. Your CRISPR target will disappear off into your cells, it will find the mutation, and it will trigger the machinery to correct that mutation back to the fully functional copy and essentially cure your disease. And these 2 diseases are the first ones to be licensed, but my expectation is that over the next 5 or 10 years, we will see more and more and more of these diseases being treated and cured, in principle, through this harnessing of a bacterial immune system. Okay, so that's a bit about bacterial immunity. What I want to turn to now is to think a little bit about broader principles of immunity. And one of the principles that we'll talk a bit about in future lectures is this idea of immunity through sacrifice. So in our immune systems as humans, many of the white blood cells that we rely on to protect ourselves are protecting us by sacrificing themselves. They find pathogens, they eat them up, and then they kill themselves, or they explode themselves to go and attack things around them. For us, as complicated multicellular organisms, that sort of makes quite a lot of sense, really, because we've got billions of cells, so losing a few to protect ourselves is quite logical. But at a single cell level, that clearly doesn't make any sense at all, and so therefore the expectation is that this kind of sacrificial immunity can't possibly exist, but actually it does. And let me explain what I mean by that. So here is a human cell. This is actually one of my cells. This is the kind of thing that human cells do. So this one is eating foreign particles, in this case a little rubber bead we've given it here, because it thinks it's foreign and it's trying to destroy that rubber bead. Okay? It does that all the time. And so right at this moment, as you're sitting here listening to me, either in this room or online, you have cells like this in your lungs, for example, that are clearing all the rubbish you're breathing in. So those of you in the room, sorry, you're in quite a crowded room here, you will be breathing in all sorts of interesting bacteria and fungi from the people sitting next to you that if you didn't deal with them, would start an infection in your lung. But that doesn't matter because cells like this are patrolling your lung and removing all that rubbish that you've just breathed in and destroying it. They will do that permanently until they sort of reach their capacity. And then when they reach their capacity, they will reach a sticky end. So here is one of those cells that we have allowed to live its full life, if you like. We have put this cell into a dish of bacteria. We've let it eat for a couple of hours, and it has filled itself. And then through a bit of microscope trickery, we can reveal the bacteria it's eaten. And what you can see in this rotating image is that that cell has eaten a lot of bacteria, perhaps 80 to 100 bacteria. It has filled itself, protecting you. It's filled itself, and now it can't eat anymore. And so now it makes the ultimate sacrifice. After realizing it's full, it sits for a little while like this. And then what you'll see in a second is that it will commit a controlled form of suicide, just like that, taking with it all the bacteria that it has swallowed in the process. And this will protect your body very effectively. It's a great system, except for if you're this cell, obviously, which means it's not so great for you. So how possibly can something like this exist in a single-celled organism? Because clearly a single-celled organism that commits suicide is not going to do very well afterwards. But there is a twist, if you like, in the microbial world that means that this kind of process does exist at a microscopic scale, and let me introduce that now. And to do that, I need to introduce you to one of my favourite microbes of all time, I have to say, which is this little chap here called Dictyostelium discoideum. And Dictyostelium is what's called, I love this, it's called a social amoeba. It makes me think there should be antisocial amoeba too, but this is definitely a social amoeba. And it's a social amoeba because it has the ability to grow as a single cell or as a cluster. So if you go out and dig in some soil and put it under a microscope, you might find Dictyostelium. It lives in sort of leaf litter out in the environment, and it survives by eating other microbes, bacteria in particular. So here he is, rumbling across the thing, and you'll see him scoff this bacteria here, and then you'll notice in a second, this one has a narrow escape because he just misses him. And so that's how they persist. They rummage around the leaf litter, eating bacteria and dividing every so often as single cells. But Dictyostelium has a remarkable trick up its sleeve. As long as there's plenty of food, it will live like this its whole life. However, if it starts to get a bit hungry, if food runs out, it realizes that it is going to be stuck, because probably if it's had no food for a little while, that means it's in an area where there is no food at all. And it needs to move, but it needs to move over a distance that it can never possibly achieve as a single cell. And so suddenly it transitions from being a single-celled to a multi-celled organism, and it does so in a really beautiful way. So this is a dish of those single-celled Dictyostelium. They have run out of food, and what you'll see over the process of several hours sped up here is they start to aggregate together. They cluster together. It's quite hypnotic, this, I find. They cluster together, forming initially kind of blobs, shapeless blobs, but then something pretty remarkable happens. Once they've formed that blob, they realize they're better as a team than individuals, and they start to behave like a multicellular slug. And in fact, we call these slugs. So you'll see out here, slugs forming in the top, and any second now, they'll start doing what slugs do and start crawling around the place, just like this. This is not an animal, right? This is a coalition of individual cells behaving as if they were a multicellular animal. And they're doing this because they're trying to search for a better location where there is food. If they don't find one fast enough, what they do instead of that is they turn into a fruiting body. So this is now looking the other way, looking vertically, and what you'll see is they give up and they turn into something which looks for all the world like a fungus, actually, with a little head up here, and they will then distribute airborne spores around. So this is microbial teamwork kind of at its absolute best. This is individual cells collaborating together. And their teamwork is so extreme that actually when they're in this multicellular state, they also use the same immune trick that we do and have a sacrificial immune system. And this is actually a relatively recent discovery, just in the last, 10 years or so. And so this is now looking down on one of those slugs at the top, and you can't quite see his head over here. And if you look down and you do some kind of molecular trickery to look at whether all the cells are the same, you see that within that multicellular slug, there are some cells, the red ones here, that are different. And these cells are sentinel cells. They are essentially the immune cells of a slug. Now remember, this slug is formed of identical cells that all came together, but at some point as they clustered together, individual cells within that have decided to adopt a different fate and take on the role of protecting the slug as a whole from infection. And they do that by eating any kind of pathogen they encounter in the process. So as the slug is crawling its way through the leaf litter, it sometimes meets bacteria that it can't eat, but actually might eat it, poisonous, dangerous bacteria. If it does, these sentinel cells will eat those bacteria, and they'll keep eating, like the ones I just showed you in humans. And then when they're full, they will move to the back of the slug, and they will drop off and kill themselves, taking with them that pathogen. And so if you have allowed a slug to crawl over a dish in which there are pathogenic bacteria, what you can see afterwards is a thing not a million miles away from a typical slug or snail trail, left behind here, all these sentinel cells, these white blood cells, if you like, with their bacteria, which have removed the bacteria from the rest of the slug that has gone on its way, now healthy and protected. So this is basically exactly the same process, but evolved in a totally different way that we are using all the time. And so in many ways, this kind of slug slime trail you see left behind Dictyostelium is exactly the equivalent of something that is much more familiar to all of us, typically when you're a teenager going on your date, which is this, the white-headed spot. So the white-headed spot is the human equivalent, because when you have a spot like this is created by the same process. What has happened here is you have had bacteria typically entering an oil gland, like in your face, in your nose or something, and starting to grow. Your immune system has realized those bacteria should not be there. In have come those cells earlier, that I showed you earlier, the neutrophils. They've eaten up the bacteria, eaten their fill, killed themselves, and now this is a full of the dead bodies of those neutrophils and all the bacteria there. And because most bacteria are sort of green or yellow, what you end up with is this fairly disgusting green or yellow thing, which is essentially a cluster of dead bacteria and dead white blood cells, just like that Dictyostelium slug. So this process of sacrificial immunity happens even at the microscopic level. And I think one of the things that we really struggle with when we think about microbes generally is because we can't see them and you can't kind of feel them, it feels a million miles away. So I want to just reflect for a second for the fact and bring this a bit closer to home. And ask the question of the audience here. I can't see you online, you have to raise your hands online. People in the audience here, who bakes their own bread occasionally or brews their own beer? Chris, it's only you. And there's a few gentle hands. Yeah, that's because you're a bit worried what I'm going to say next. Okay. So if you've ever done your own beer, your own fermentation of anything, then you actually have been witness to one of the most amazing immune battles in the microbial world that we know of. Because you will have used, I hope at least, to create that yeast of some form. So typically if you make bread, right, you buy a packet of yeast from your supermarket and you sprinkle it on. If you do sourdough, it's kind of slightly different, but we'll go there later. So assume you've used yeast. This is Saccharomyces cerevisiae, the brewer's yeast, the same one that we use for bread making. It's a fungus, okay, it's a yeast. And the whole process of making bread or beer obviously relies in this guy growing really, really, really fast and generating that kind of light, fluffy bread that you're looking for. When it's doing that, that moment when it's bubbling away in your kitchen, it is undergoing an enormous immune battle because it turns out that every domesticated strain of yeast that we have in the world is infected with a virus called LA virus. This is what it looks like. This is it's called a cryo-EM image, it's an electron microscope reconstruction. This virus is embedded in the genome of Saccharomyces cerevisiae, and it's present in all domestic strains, most wild strains, but not all. There are some strains out there that are protected from this, but as far as we know, every yeast anywhere in the world that's in a kitchen has got this virus in it. And this virus is potentially able to kill the yeast, but obviously when you make bread, it's not killing the yeast because the yeast is surviving and growing. And that is because the yeast has an immune system too that fights this virus off. And you can sort of see that in this example here. So this is a wild-type yeast. I'm not sure how well this works. This is a single yeast cell, which is kind of highlighted in the white dotted line here. And what we can see in that yeast cell in blue is the yeast DNA. Okay, this is the yeast nucleus, which is what makes the yeast a yeast. But in red is DNA that's not yeast DNA. This is the virus genome, okay? And what you can see, even in a wild-type, healthy, happy yeast that you've got from your supermarket, you can see the virus is growing quite faintly. However, if you go through that yeast genome and you look for genes that the yeast might be using to protect itself from it, and you disable those genes one by one, you can create a yeast that is highly immunocompromised. It's the equivalent, if you like, of a human with HIV or organ transplantation suppression or something like that. It is unable to function normally in an immune system, and those yeast, as you can see here, suffer from a massive abundance of virus. And in fact, they do not grow well and usually die out. So if this was to happen in your bread creation, you wouldn't have very nice fluffy bread. In fact, the yeast wouldn't grow at all. Because the yeast has a multiple sets of genes that protect it from virus, even though it's latently infected, it actually grows quite well. And to me, the most amazing thing is when people look at what those genes are, some of the genes the yeast uses to protect itself from a virus are exactly the same as the ones we use. So for example, here is this is now as a protein, three-dimensional protein, that the yeast uses. It's called PABC, it's a bit of a clunky name, and it's a protein that sticks to that viral DNA and stops it creating new viruses. And this protein is almost identical to one, I know you're not all protein aficionados, but believe me, it's almost identical to one that we have in humans. And in fact, it turns out that this protein in humans is really important for fighting off things like COVID-19. So the next time you're making bread, I want you to look into that pot and think, crikey, right at that moment, that poor yeast is fighting off all that viral infestation in exactly the same way as I fought off COVID-19 or flu or whatever else you've had recently, with exactly the same sorts of proteins. So there are all sorts of different microbial immune systems. There are all sorts of different processes. There's a remarkable synergy between them. And so in the last sort of few minutes, what I want to think about a little bit is what this means for our understanding of kind of microbial ecology and our place in the universe, if you like, which sounds very dramatic. And I often like to use this sort of quote here, which is from, if you know, a Jonathan Swift poem, so he of Gulliver's Travels. He wrote this poem, "Big fleas have little fleas upon their backs to bite 'em, and little fleas have lesser fleas, and so ad infinitum," which I quite like. In other words, wherever you are in the food chain, there is always someone who eats you and someone that you eat. And it doesn't matter how high or how low. And that is true also for infections, and it is true for immunity generally. And I want to kind of illustrate that by thinking about this process of microbial immunity and microbial pathogens. And I want to think about the smallest type of microbe that we know of, which is the virus, right? So viruses, as people may know, there's often a debate about whether a virus is even alive, because viruses, by definition, can only replicate in the presence of a host. Okay? So if you have a vial in a lab of COVID-19 or HIV or anything else, it is completely inactive in a way that it's not true for bacteria. Bacteria will kind of grow in their own kind of vial. But a virus is inactive until you put it in a host cell. It requires the host cell to replicate. So you might expect, therefore, that that's the kind of smallest possible type of microbe, and therefore the viruses, by definition, cannot have pathogens of themselves. But you'd be wrong. So here is a typical virus. This is SARS-CoV-2, the causative agent of COVID-19. It is tiny. It is very simple, actually. It is a small number, a handful of genes, essentially, inside a shell. So what COVID-19 has in its genome are the genes to replicate its own genome and to produce the couple of proteins it needs to encapsulate itself, and that's it. It's like the ultimately trimmed down version of a virus. And it's very effective, but it is completely reliant on the host. But fairly recently, about 20 or so years ago, people discovered there are a class of viruses that are much bigger and more complicated than that. And this is an example of one. This is called virus, and it's a different sort of imaging, but it is at the same scale. So this guy is about 5 times, 6 times as wide as COVID-19, or if you do it in volume times, we're talking about sort of 100 times bigger. It's a real giant of a virus. And in fact, it's so giant that it turns out that people had inadvertently discovered this back in 1982, but they thought it must be a bacteria because it's too big. And one of my favourite stories about this, it was discovered in a cooling tower in Sheffield, which is definitely not the kind of exotic place you expect to find a new fundamental species, but there you go. So if you ever travel out, you see this big cooling tower, don't see so many anymore, happily, where the steam is coming out the top. And because they are permanently wet and permanently at a very similar temperature, they're actually an interesting environment for new microbes to exist. And so people used to go down and kind of scavenge stuff off cooling tower insides and look what's living there. And they saw this under a microscope image, and they said, it must be a kind of weird bacteria. We'll classify it, we'll tuck it away forever. And it sat in an archive for over 20 years. And then much, much later, people realized, actually, this is not a bacteria, this is a giant virus. And it turns out to be the first of what is now quite a big family of giant viruses. So they're giant in size, they're also giant in complexity. So unlike things like HIV, which has 9 proteins or so, or COVID-19, this guy has quite a big genome and quite a big set of proteins, including proteins that it uses to do some of the functions that SARS-CoV-2 relies on the human cell to do. So this virus, for example, can't make any of its own protein. It uses your cells to do that. It can't do lots of the fundamental functions that it would require to live. This virus, on the other hand, encodes some of its own machinery to make proteins. So when it gets into a cell, it hijacks some of the cell's machinery, but it also produces some of its own. So people often think of these giant viruses as being a kind of transitionary life form between something that's not really alive, like this, and something that's definitely alive, like a bacteria. It's sort of on the cusp. So why am I telling you this in a lecture about immunity? I'm telling you this because this is a giant virus, and this virus, because it's so big, has viruses itself. And so just a few years ago, people discovered when studying this that if you look closely, when a amoebae virus, which is this guy here, infects a cell, you sometimes see these little tiny viruses associated with it. And it turns out that indeed this virus is a virus of a virus. So this guy is called amoebae virus, and it's short for microbe mimicking, because people thought it looked like a bacteria. And this little chap here is called Sputnik, because for people old enough listening to this, Sputnik was the first satellite. Chris, I'm glad you're hosting today, given that we have a bit of a space theme here. Sputnik was the first satellite in orbit, and this is a satellite virus of this one because it's seen kind of closely in the orbit of this. Biologists are not very creative. We just go, that looks a bit like an orbit, we'll call it Sputnik. So Sputnik sits here. And it turns out that Sputnik is a virus that can only survive when it's present in a cell with amoebae virus. Amoebae virus is a virus, so it can only survive when it's inside a cell itself. So what you have here is a kind of a sort of hierarchy of dependencies. And so now we're in a situation where you have if you like, a virus of a virus. So you might therefore think, well, in that case, if there's a virus of a virus, is it possible that this virus has got some level of immunity to this virus here? And it turns out that that is true. So here you can have 2 images, if you like, which hopefully you can just about see here. So in this case, what you see on this side, let's get started over here. This is a host cell that's infected with amoebae virus. And here is the amoebae virus. You can see them scattered around. In this case over here, what you can see also is a host cell with the amoebae virus. Have I got a little pointer up here? No, I don't. So you can see amoebae virus, for example, right up there. But you can also see these little tiny dots. These are the Sputnik virus. Okay? And the difference here is that in this case, Sputnik is growing on amoebae virus. Here, the amoebae virus has fought off the Sputnik virus. And it's fought it off using an immune system that is very closely related to that CRISPR system that bacteria have. So here we have a virus fighting off another virus using an immune system that we saw in a bacteria, which I think is super cute. And it turns out, in a really remarkable way, that that process of immune fighting, if you like, has been harnessed to another level by some other microbes. So, amoebae virus infects a variety of single-celled microbes, eukaryotic microbes, like protists, like amoeba, for example. This is one of those single-celled microbes that suffers from giant virus infections. And I love this particular chap. So this is found in kind of pond water. It swims using these little tails. Here I don't have a movie, sadly. And I love this one because its Latin name is cafeteria. What a great name that is. So you can officially say, if you work on this, I'm just working in the cafeteria. So this cafeteria gets infected by giant viruses. And it's trouble. You don't want to be infected by giant viruses. So what cafeteria has done is it has embedded in its own genome one of those satellite viruses like Sputnik that sits inside its genome. And now, if one of those giant viruses like amoebae virus tries to infect cafeteria, it wakes up the Sputnik virus and wipes out the infecting giant virus. So it is harnessing a virus of a virus to protect itself against the giant virus, which I think is really, really cute. So let me finish by just putting all that together and reflecting on what this might all mean for us as humans. And I'm going to pick on some people in the audience, we'll see if anyone's here. Who's a contact lens wearer? I love the hands get even lower every time I ask a question. Like, maybe. Okay, and I won't pick on people to answer this, but when you deal with your contact lenses and you're cleaning them, do you clean them in contact lens fluid? Please nod. Do you ever clean them in running water? Please shake your head. And the reason, or I'm sure if you're a contact lens wearer, you clearly see on it, don't wash this in water or whatever else, use proper sterile contact lens fluid, is because if you're a contact lens wearer in particular, normal water out there can contain a little beast called acanthamoeba that is quite common in water. Acanthamoeba rummages around the environment, it's happy out in the environment, it eats bacteria, all these kind of things, but unfortunately, if you're a contact lens wearer and you get acanthamoeba on the inside of your contact lens and stick it on your eye, acanthamoeba doesn't have any bacteria to eat there, so it starts nibbling away on your eyeball instead, and that is not great. And you can get a disease called acanthamoeba keratitis, which I have not got pictures of because it's quite grim. So this is one of the reasons you have to really look after your contact lenses. So acanthamoeba keratitis is a problem for humans. First pathogen, right? But now, as you've learned hopefully during this lecture, this pathogen also has pathogens. So if you've got acanthamoeba, that acanthamoeba itself might also be having a bad day, because it, for example, might have within it an intracellular pathogenic bacterium like this one. This is Mycobacterium marinum. It's related to tuberculosis in humans, and it infects all sorts of things, including acanthamoeba. So your acanthamoeba that's eating your eyeball might itself be eaten from the inside by Mycobacterium. It might also have a giant virus, because this is one of the species that giant viruses infect. So it might be having a really bad day when it's being eaten alive by bacteria and giant viruses. But don't panic, because it's possible, if you're a lucky acanthamoeba, that at the same time, you have also got a bacteriophage that's killing your mycobacteria that was trying to kill your acanthamoeba that was trying to kill your eyeball, or a satellite virus that's killing your giant virus that was trying to kill your acanthamoeba that was trying to kill your eyeball. And so next time you put your contact lens on and you think, this is a bit dodgy, I shouldn't have washed it in water, reflect on the fact that there is an entire hierarchy of things eating each other on your eyeball, and hopefully the right one will win and protect your eye for tomorrow. Thank you very much indeed.